Cool. Thanks everyone, my name is Ben. Today I'm going to present to you a paper from Michael Hofreiter's group at the University of Leipzig titled Nuclear Gene Indicates Coat Color Polymorphism in Mammoths. So this group is primarily interested in studying the role of the melanocortin receptor in mammoths. And a little bit of background on the melanocortin receptor is it's the primary receptor for hair color in mammals. So MC1R, the melanocortin receptor, is a G protein coupled receptor that when stimulated produces cyclic AMP, the downstream signaling result of which leads to the production of brown or black melanin, but impaired deficiency or impaired MC1R gene leads to the brown or red hair color that is seen in most mammals. In particular, the question they wanted to ask is, did mammoths have different hair colors? There's some circumstantial evidence from hair samples provided that mammoths may have had different hair colors, but they had no prior evidence of the precise genes that controlled mammoth hair color. But really, who cares about mammal fur? I mean, these animals have been extinct for tens of thousands of years, and that's really not the primary point of the paper. The primary point of the paper is they now show the feasibility of possible study of extinct gene function, or gene function in extinct species. So essentially what this group did is they took 4 mammoth bones and ground up the bones and extracted the DNA, and then ran some PCR with some primers, extracted some primer fragments from Asian and African elephants, and then sequenced the melanocortin receptor that they were able to isolate. Essentially what they found is that there were 2 different alleles in mammoths. One that they titled allele 1, and the other that had allele 2 with a couple of different amino acid substitutions in them. They wanted to ask, how do these polymorphisms affect MC1R activity? So they took this mammoth MC1R gene that they cloned and they transfected it into COS7 cells, and they ran an assay to look at cyclic AMP production upon stimulation. Essentially what they found is that there's these 2 different alleles. One had much reduced activity, allele 2, and this is entirely attributing to an arginine to cysteine mutation. So essentially what they've shown is that mammoth gene sequencing proves the feasibility of studying gene function in extinct species, not only for mammoths, but for other extinct species long ago. And also for humans. I think the real importance of this paper is that we now have a method for studying human evolution and gene function in our human ancestors. So in conclusion, PCR amplification was used to identify genetic polymorphisms in the mammoth gene for MC1R. Functional analysis of the gene shows reduced activity. Reduced activity correlates with red and brown hair in humans, thus mammoths may have had red and brown hair as well, and that this technique could be used to study gene activity in other extinct species. Thank you.